Hello, everyone, and welcome to our webinar today, the Australian Early Phase Biosimilars Clinical Trial Landscape, How Agilex Biolabs and Nucleus Network Work Together. I'm Arsalan Arif, the publisher of Endpoints News, and it's my pleasure to have Kurt Sales, the Director of Immunoassay at Agilex Biolabs, and Dr. Paul Griffin, Medical Director and a Principal Investigator of Nucleus Network with us today. We've got a presentation from Kurt, approximately 30 minutes, followed by a Q&A session with both Kurt and Dr. Paul Griffin. Now, I encourage the audience to submit questions using the Q&A button below. I'm going to get to as many as possible, but if we don't get to your question, someone from the Agilex Biolabs team will reach out to you directly. And now, without further ado, let's turn it over to Kurt.
Thanks very much, Arslan, and very good day to all the viewers out there, and thanks for tuning in for this webinar from Agilex and Nucleus Network. Really, this little webinar will focus on the clinical trial landscape in Australia, what that looks like, a little bit about Agilex Biolabs, our capabilities, how we fit into the clinical trials network, and specifically really how we work with phase 1 units such as Nucleus Network in order to deliver biosimilar programs. So really, the Australian advantage is underpinned by three aspects, and these are quality, speed, and cost. If we firstly focus on quality, really the quality of our medical system and facilities are second to none. Most of our key opinion leaders have not only trained in Australia, but also extensively overseas. They're well connected with international colleagues and at the forefront of medical technologies. Data from our clinical trials conducted in Australia is also compliant with regulatory agencies, including the FDA, EMA, MFDS, and TFDA. In terms of speed, our regulatory process is extremely streamlined, which allows a sponsor to get their study into the clinic faster. A full CMC package, for example, is not required, but really instead a protocol, informed consent, insurance certificate, and investigator brochure containing all the relevant preclinical data or supportive data is all that the ethics committee is really required to lodge a decision. And documents such as patient consent, really, the consent forms don't usually require translation. So this enables first-in-man studies to be conducted in a rapid startup of about 4 to 6 weeks. And typically, especially even with contracting with Agilex, contracts are reviewed very efficiently, and that allows fast startup of the bioanalysis. And as a laboratory, we can also do quick and fast sample turnaround. In terms of cost, biotech companies may be eligible for a research and development rebate, which is about 43.5%. That's really paid at the end of each financial year. And that is on all of the activities associated with the clinical trial, not necessarily just the bioanalysis, but the clinical trial itself, any CMC or manufacturing that might have occurred, and so on. In order to do this, companies just need to establish a local entity. This is really inexpensive. We can provide some information on how to go about doing this. It takes a couple of weeks. There's substantial cost savings, and especially for small biotech companies that are in the sort of burning rather than earning phase, this allows the cash to just spread that much faster on these studies. And generally, we're about 15 to 20% lower in Australia than in USA and the Western Europe. I will apologize in advance. We're currently busy expanding our laboratory, so there might be a little bit of background noise occasionally, but unfortunately the contractors are on site, so there's not much I can do about that this morning. I'll focus a little bit about Agilex Biolabs. We're the leading bioanalytical laboratory in Australia with more than 20 years experience, and we really focus on delivering market-leading service of the highest bioanalytical quality internationally. Cumulatively, we've focused on delivering more than 350 projects over the past 20 years. Our repeat rate is more than 85%. And we work globally with all of the CROs internationally, locally, and our clients have a broad global spectrum. Our service lines include tandem mass spectrometry, so LC-MS bioanalysis, and also immunoassay and immunobiology analysis. This allows us to focus on PK, immunogenicity, and biomarker delivery. We're a customer-focused laboratory. We focus on data integrity. All of our data is according to OECD GLP quality systems. We're also associated, the NATA local NATA accreditation of ISO 17025 and GLP, and we focus really on timeliness and speed. Our laboratory of more than 60 analytical scientists, many of these are PhD level scientists that are specialty chemists in their own right. This allows us to deliver LC-MS, small molecule bioanalysis, large molecule immunoassay bioanalysis, and also antibody drug conjugates and biomarkers. In terms of our biosimilar capabilities, for biosimilar approval by regulatory authorities, comparison tests against a reference innovative product is required, and this is required for safety, purity, potency, stability, and also evaluation of immunogenicity. And so the bioanalytical assays required to support these trials include pharmacokinetics, pharmacodynamic biomarkers, immunogenicity, anti-drug antibody assays, and also neutralizing antibody assays. At Agilex Biolabs, we've developed a variety of off-the-shelf PK and immunogenicity assays over the past couple of years, several of which are highlighted on this slide. And this allows for time and cost-effective setup of the required assays for your trial. We have strong biosimilar experience across different compounds and assays, and this reduces the risk to the clinical trial because it enables us to allow fast setup of assays. Our scientific expertise is second to none, and every project that we deal with is unique, and we tailor the project to suit the sponsor. In terms of immunoassay formats and instrumentation, we can deal with a variety of different platforms in immunoassay, for example, ELISA by fluorescence, chemiluminescence, or colorimetric assays. We can deal with electrochemiluminescent assays by mesoscale discovery. More importantly, we've recently introduced the Gyrolab Explore, which is an automated nanoliter, automated microassay immunoassay platform. And we can also conduct small molecule bioanalysis by tandem spectrometry to support biosimilars such as insulin glargine, etc. Our instrumentation goes through a Watson-LIMS system, which is all validated and under change control, and this allows for data to be available pretty quickly. In terms of PK assays, we can either develop the assay de novo from scratch for a client for a specific or new biosimilar coming off patent, or we can transfer a method in from the client's lab or another laboratory. And we can utilize multiple different instrument platforms, as I mentioned in the previous slide, and this we can tailor to the specific requirements of the molecules. Some molecules require highly sensitive assays and others less. And this allows us to also measure free or total drug in serum or plasma. Immunogenicity is really important, especially when it comes to phase 3 biosimilar trials, where monitoring it becomes paramount to determine whether the drug is still effective over a longer time period and whether the patient is developing any anti-drug antibodies, which might reduce the efficacy of the compound. So we can develop a single ADA assay, which is a screening, confirmatory, and titer assay to determine the ADAs. And we use a tiered approach as outlined by the FDA bioanalytical guidance. More importantly, as a laboratory, we've got substantial expertise in non-standard assay formats to deal with drug tolerance, specific for phase 3 trials, where the dose is substantially higher than in a healthy volunteer study, or the dose plateaus over a long period of time due to constant dosing. For these assays, sensitive assays to deal with drug tolerance and target engagement is paramount. And so we've developed a variety of non-standard formats for our biosimilars over the past couple of years, and these include acid dissociation, affinity capture elution, or ACE, precipitation and acid dissociation, or PANDA, and also solid phase extraction and acid dissociation, or SPEED. And these really mitigate interferences and help us meet sensitivity and drug tolerance. And all of our ADA statistical analysis is applied for cut point assessment using the latest industry guidelines. Following on from just the standard immunogenicity, we can also develop neutralizing antibody assays, and these then determine whether the ADA that is developed by the patient has a neutralizing effect and would then wipe out the efficacy of that drug. And we can do these either by a competitive ligand binding or a cell-based assay solution. Some clients are interested in determining whether the biomarker actually has that desired effect. For example, omalizumab, which targets IgE. Some clients would want to have a free or total IgE assay as an efficacy endpoint. And these are assays that we can actually add on as well to our bioanalysis. Just a little bit about the JARA-LAB Explore and why it's such a fantastic instrument and why we've installed it in Agilex Biolabs in order to deal with biosimilar studies in particular. Biosimilar studies utilize quite a large population, especially in phase 3 studies, and often fast turnaround of data is required. So in order to churn around thousands of samples, you need something that's a little bit more high-tech and automated. Enter the JARA-LAB Explore, which is a semi-automated miniaturized system. It uses nanoliter amounts of sample and volumes and has a very fast runtime. And this slide just gives you an idea of what you could do in a working week. Down below, if we ran the JARA-LAB Explore versus an MSD or an ELISA, we could churn through, for example, 1,125 samples in a week just using a JARA-LAB Explore with one operator versus 300 samples using a classic plate-based assay on an MSD or ELISA. So the JARA-LAB Explore really allows us to be able to turn the clinical trial around a lot faster to provide the PK statisticians with the data or to provide safety review committees with the data that much faster to enable phase 1 units such as Nucleus Network to be able to deliver in a faster time frame as well. I'm just going to focus very briefly on some of the performance characteristics of these assays because I think they are fantastic and they're so precise and sensitive. And I'm just giving a little example of what the classic range would look like for on the left-hand side, a JARA-LAB versus an ELISA, and on the right-hand side, a JARA-LAB versus an MSD, just to highlight, the advantages of this platform. So on the left-hand side, you can see on the x-axis, the JARA-LAB Explore has this fantastic logarithmic dynamic range. And the advantage of that is that you're highly unlikely to ever having to dilute a sample because it's out of range. And this translates to speed and also cost effectiveness for the client. In terms of sensitivity, the gold standard in sensitivity was always the MSD. The JARA-LAB Explore is actually comparable in terms of sensitivity to the MSD. It just utilizes a slightly different chemistry, that being fluorescence. So one of the key processes to de-risk the clinical trial is timeliness. This overview basically gives an idea of what is generally required to get the bioanalysis set up. Once the phase 1 unit has been contracted and the CRO, more often than not, the PK assays and the immunogenicity are sometimes a little bit of an afterthought. To bring this forward and to provide a context, it sometimes takes up to 6 weeks to develop a PK or immunogenicity assay from scratch for a very complicated molecule, and then an additional 4 weeks to validate the assay according to bioanalytical guidance documentation. So really, you're looking at about 12 weeks for a bioanalysis package, and that needs to be put in place prior to first patient, first dose. And this is what I'm highlighting in this slide. And this allows us to have everything aligned for first patient, first dose dosage, and therefore sample delivery from the phase 1 units, such as Nucleus Network. In terms of the clinical trial ecosystem, this is where we fit in. We are agnostic and work with all CROs, specialty units like Nucleus Network for biosimilars. They're the largest clinical trials phase 1 unit with footprints in both Australia and the US to be able to deliver on this type of trial. And we are the largest bioanalytical lab with the greatest expertise in not only PK, but immunogenicity. And so we fit into this landscape together with all of the other providers to provide the client with a full package for the biosimilar clinical trial. I hope that you found some of this data to be informative and interesting. I'm always happy to answer some questions offline, yeah, we'd be happy to take some further questions from you. Thank you.
All right. Thank you very much, Kurt. I appreciate that. That was very snappy and a lot of great information over there. I've got some questions. We've got some questions from the audience. We've got a little bit of time here. Kurt, let me actually just start with you. How many phase one units does Agilex Biolabs work with? And how does the collaboration between Agilex and Nucleus Network benefit the trial sponsor?
So Agilex is pretty agnostic with who we work with. Because we're the largest lab, we work with everybody. And every phase one unit has a certain specialty. So the types of clinical trials that we as a bioanalytical laboratory get from the phase one units is inherently different. And it's driven by, whether it's a healthy volunteer study or a patient-specific study, etc. So we work with everybody. And in terms of biosimilar studies, we work with everybody in Australia as well as New Zealand. And we work with Nucleus Network, for example, because they are the largest provider for biosimilar studies in Australia. They've been around for as long as we have been around, pretty much. We work quite closely together during the study in order to provide a seamless back-to-back service for the client. And that is, once the subjects or the patients have been dosed in the phase one unit, the bloods are collected and they are rapidly shipped over to Agilex Biolabs for analysis. So the communication between the two companies is second to none, and that just enables us to deliver a higher quality product for the client in biosimilars. And that's why we work so closely together and have such a good working relationship.
Okay. And Dr. Paul Griffin, same question to you. I mean, how does that collaboration benefit the trial sponsor?
Yeah, thank you. I mean, great presentation and really glad I could be part of the webinar. So obviously, Australia's largest phase one unit working with the leading bioanalytical laboratory makes sense. I guess with these sort of trials, we need the highest quality of data from the participants and also the highest quality of data in terms of the results of the assays. And, we know that both of those things are interdependent. And so to generate good data from the assays, we need good specimens collected at the right time. And obviously we need good accompanying clinical data with that. And that's why, the leading bioanalytical lab partnering with the largest phase one unit makes perfect sense, because we work so well together and make sure that, end to end from start to finish of the clinical trial, everything is done of the highest quality.
All right. Let's talk about Nucleus Network a little bit. I mean, tell me about some of the attributes over there. What's going to make Nucleus Network a good option for biosimilar studies?
So we are Australia's largest phase one unit. So we have a footprint in 2 states in Australia. Nucleus has been around for nearly 20 years down in Victoria, and then acquired Queensland's largest phase one unit, Q-Pharm, some years ago, making it definitely the largest phase one company in Australia. We've done over around 800 early phase trials, a large number of those first in human, and a lot of biosimilar studies. And we have a lot of expertise as well as experience. So our Chief Medical Officer is an oncologist by training. I'm an infectious diseases physician. We have clinical pharmacologists. So we have a lot of expertise in this area. And, clearly quality is the main thing that we focus on. And having done so many studies and undergone a number of audits, we certainly are confident of our quality with all of our clinical trials.
Okay. Well, let me ask you, as a principal investigator over there, talk to us about some of the specific challenges around running biosimilar trials. Can you talk to us about your top sort of 1 or 2 challenges as you see as a PI?
Yeah, look, it's hard to bring that down to 1 or 2. Often they're fairly, complex products with interesting mechanisms of action. And so giving those to healthy volunteers can be a little bit of a challenge at times. But I think that's where our experience really comes in terms of being able to assess and ameliorate those risks, make sure we, appropriately consent all our volunteers, that we do everything we can to make sure the volunteers are safe throughout. And if they do experience any adverse events, then we can manage them appropriately. And of course, if the trials aren't done of sufficient quality, then obviously the licensing or the subsequent approval of the product is potentially not able to happen. So that's where I guess, the attention to detail and quality that we have really lends itself to doing these sort of trials and overcomes that problem as well.
Okay, well, let me try to ask you a little bit more of a specific question then, okay, about biosimilar trials. How are you recruiting for participants right now in the midst of a pandemic?
Yeah, great question. So we know clinical trial participation has suffered a little bit in the context of the pandemic for a host of reasons. Very early on, we brought in some very specific interventions to make sure all of our staff and volunteers are safe throughout. And that's allowed us to continue operating, not only here in Queensland, where we have excellent control of the pandemic more broadly, but even in Victoria, where they've had their obvious struggles with this. We've been able to keep doing not only our COVID-related clinical trials, but all of our clinical trials, including biosimilars. So, I think it's about ensuring safety, but then also having a recruitment team with a lot of experience and expertise and a great deal of success recruiting people for these sorts of trials. And I think also in Australia, we're fortunate to have a population that is very able and willing to participate in these sorts of trials because they understand just how important they are.
Right, right. So right now you're telling me you really haven't had any kind of issue recruiting people in the midst of a pandemic.
No issues really. I mean, a lot of work has gone into making sure we can do that safely and successfully, and that's been the case. And so overall, our recruitment hasn't really been compromised at all.
Are there any specifics you could share with our audience on any of those procedures that you're taking to ensure that? Any kind of tips you could offer our audience on that? I mean, this is a topic of just utmost importance.
I think just having a really comprehensive plan. So pre-screening of everyone who's coming on site, obviously limiting any unnecessary attendances. So remote monitoring, for example, temperature screening upon arrival. Everyone wears masks, all staff are in scrubs. We're maintaining social distancing throughout of staff as well as volunteers, paying great attention to hand hygiene. And if any staff or participant exhibits any potentially compatible symptoms, then we remove those people from our site and get them quickly tested to see if it's been COVID. So as I say, a very comprehensive plan that has been rigorously implemented.
That does sound complete. Okay, well, I'm just going to wrap up this snap webinar with one question over here for you, Dr. Griffin. Is Nucleus Network able to conduct trials for patients as well as healthy participants?
Absolutely. We can conduct all clinical trials, basically. I mean, as Australia's largest phase 1 clinical trial company, our focus is often on early phase trials, but we can certainly support our internal investigators conducting later phase trials or even work collaboratively with external investigators in clinical environments to help them with their later phase trials as well. So yes, basically we do have that capacity and we do a lot of that work.
All right. Okay. Well, that's the time that we have for today. I want to thank the entire audience for joining us. There's several questions that we did not get to, but the Agilex Biolabs team is going to follow up with you directly on that. I'd like to thank Agilex Biolabs for sponsoring today's session and Kurt Sales and Dr. Paul Griffin for their time and expertise today. Thank you, Kurt, for your presentation. I really enjoyed it. I hope to see everyone back here for another Endpoints webinar soon. Have a wonderful rest of your day.
Thank you. Thanks. Bye-bye.